Spider-Man, Spider-Man, who's the greatest Spider-Man? Now, you've probably heard of Spider-Man, but you know he's also a made-up character. But did you know that scientists have been able to create a spider goat? A goat which produces spider silk in its milk. Now, that sounds really strange, but with genetic engineering, such feats are now possible. Now, do remind me to show you this video when we're back in class. But first, let's take a look at what genetic engineering is all about. So in this video, we'll be looking at what is genetic engineering, what are some examples, and we'll look at how genetic engineering is actually carried out. And in class, when you do your presentations, we'll look at what are the social and ethical implications of genetic engineering. So what exactly is genetic engineering? Well, it refers to a technique that is used to transfer genes from one organism to another. So in the case of the spider goats, what we did was we transferred a gene from the spider to the goat, right? So this goat now is known as a transgenic organism. A transgenic organism refers to an organism that has acquired foreign genes from another organism, right, through an artificial method like genetic engineering. Now, this transgenic organism may acquire foreign genes from another organism of a different species, or it may actually acquire foreign genes from another organism of the same species, as we shall see later on. Now, just a side point, that the reason we are able to transfer genes between organisms is because the genetic code is universal. Now, what that means is that in all organisms, whether you're a spider or human being or goat, all organisms use the same processes to express genes. Or put it in a different way, all organisms use the same processes to decode the gene into a polypeptide. Right? To convert gene to polypeptide, all organisms use the same processes of transcription and translation. So if I put a spider gene into the goat cells are able to interpret that gene. They're able to still decode that gene because they use the same processes as the spider cells. Right? So because the genetic code is the same in all organisms, then genetic engineering makes sense. I can shift genes from one organism to a different organism. Alright, so it sounds really cool, right? We can shift genes from one organism to another. But how exactly is it done? What are the steps involved? Okay, so let's take the example of trying to transfer the spider silk making gene from the spider to the goat. Okay, so the first step is that I need to actually isolate my desired gene. In this case, my desired gene is the spider silk making gene. So I need to isolate it. I need to take it out from the cells of the spider. And this process involves what is called restriction enzymes, which we'll take a look at later. So imagine I take out the gene. I'm going to take out the gene from the spider. This is the gene that makes spider silk. Now the next step is I need to insert this gene, this desired gene, into a vector. So we'll take a look at what vectors are later. Okay, but it's just something that helps to carry that gene. And the next step is I need to then insert this vector. Right? I use the vector to insert the desired gene into the recipient organism. So in this case, I want to use the vector to insert the gene into cells of the goat. Right? So I insert it into the cells of the goat. And at the end of the day, what I will get is that this gene, the spider silk making gene, is then expressed in my recipient organism, in this case the goat. So then the goat will be able to produce silk. Now for the rest of this video, we're going to take a look at a number of case studies. Now the main case study that you need to be familiar with is the use of genetic engineering to make transgenic bacteria that produce human insulin. Now we've learned about insulin before. We know that it is important in helping to regulate blood glucose concentration. Now people with diabetes type 1 hopefully remember that they are not able to produce insulin. So how do we help such people? Well, normally what we can do is they need insulin injections, right? And this insulin we can normally obtain them from animals like pigs, But there are certain problems with this. So if you get insulin from animals, then if you inject it into a person, it might cause allergies, might cause contamination. It's quite costly, and some religions might be uncomfortable with pig insulin in them. So what solution have we come up with? Well, we've come up with a solution where we use genetic engineering to create transgenic bacteria that contain the human insulin gene. And so these transgenic bacteria are able to produce insulin for us, human insulin, which I can then extract out and I can then use it to help people with diabetes type 1. Alright, so how do we insert the human insulin gene into a bacteria? Well, the first step is that we need to actually isolate the human insulin gene. So we need to take the human insulin gene out of humans, right? Take it out of our human cells. And what we'll do next is we'll insert the human insulin gene into a vector. In this case, the vector is a bacterial plasmid, which we'll take a look at what that is in just a while. Then what I do next is I insert this vector, I insert the plasmid into the bacteria. Okay, here's my bacteria. I insert it inside. So now the bacteria has the vector, which has the human insulin gene. So the bacteria is now transgenic. It's able to express the human insulin gene and to produce insulin for me. Okay, so let's take a closer look now at each step of the process. So first step was to isolate or to take out that human insulin gene. How do I do that? First, I need to find out which chromosome is that insulin gene on. So imagine I found that out. So here's my chromosome that contains the insulin gene, here shown in yellow. How do I then cut out the insulin gene from this very long chromosome? Well, I cut it out by using what is called a restriction enzyme. So I will mix the chromosome together with a restriction enzyme, which I'm going to just draw out like that. I mix it together with the restriction enzyme, and the restriction enzyme will act on certain specific sites along the chromosome. So it'll act at these particular DNA sequences, and it'll cut at these particular sequences. So when I cut the gene out using the restriction enzyme, I will produce a DNA fragment, and this DNA fragment contains the insulin gene, and at the sites, at each end of the insulin gene, there will be what is called sticky ends. Okay, later I'll explain what sticky ends do. So just a further note on restriction enzymes. Restriction enzymes act a little bit like scissors. So they recognize certain sites and they cut at those sites. In this case, the example of a restriction enzyme would be something like EcoRI. What it does is that it recognizes this sequence, TTAA, and it cuts. When it sees that, it just cuts it in this way to produce a sticky end. So this sticky end here, TTAA, is actually just a single-stranded protrusion. So this is a sticky end. And this protrusion is very helpful because if it can find a matching sticky end, then it will actually join back. And we'll see that that's really important in just a while. Alright, so step 2 was to insert the human insulin gene into a vector. And in this case, our vector is a bacterial plasmid. So what is a plasmid? Well, a plasmid is simply a circular ring of DNA that is found in bacteria. So bacteria do not have a nucleus. However, they have some circular rings of DNA called plasmids. Right, so we are going to make use of this as our vectors. So here's a bacterial plasmid. It looks something like this, a circular ring of DNA. Next, what we'll do is we're going to cut open that plasmid using the same restriction enzyme. It's important that we use the same restriction enzyme because this will produce sticky ends, as you can see here, that are complementary to the sticky ends of that DNA fragment containing the insulin gene. So these sticky ends here are complementary to those that we produced at the ends of the insulin gene because we use the same restriction enzyme. So now in my next step, what I do is that I take that insulin gene with the sticky ends and I mix it together with the plasmid with sticky ends, and the insulin gene will then bind to the plasmid due to that complementary base pairing between the sticky ends. So you can see here that the sticky ends have started to bind together due to complementary base pairing. And it's held together by the hydrogen bonds between the complementary bases, right? However, these hydrogen bonds are not entirely strong enough. They're not able to hold this insulin gene inside the plasmid permanently. So we need to use an enzyme called DNA ligase to seal the gene and the plasmid together. So DNA ligase is a bit like a glue, right? So the restriction enzyme was something like a scissors, and then the DNA ligase is a bit like a glue that sticks them together. Alright, so now we have a plasmid that contains the human insulin gene. Now this plasmid that contains the human insulin gene can be called a recombinant plasmid. And it's called recombinant plasmid because it contains DNA from two different organisms. In this case, it contains DNA from the bacteria and DNA from humans. And what we do next is that we then take this recombinant plasmid and we insert it back into the bacteria. So here's my recombinant plasmid. It's recombinant because it contains the human insulin gene as well as the bacterial DNA. And here is my bacteria, and I'm going to insert that recombinant plasmid back into the bacteria. And as a result, I produce a transgenic bacterium because it's transgenic because it has acquired foreign genes from a different organism, humans, through artificial methods. Alright, so now I have my transgenic bacteria. It contains the human insulin gene, it's able to express it and produce insulin. And I can grow the bacteria in these large containers called fermenters. And after a while, they'll produce all the insulin. What I need to do then is to extract and to purify the insulin to be used. Now, as a bit of extra information, you might be wondering, how did we insert that plasmid back into the bacteria? I mean, the bacteria has like a cell membrane and a cell wall. How do I put it back in? Well, what people do is that there are two main methods they usually use. They use either the heat shock method or the electric shock method. So you basically mix the bacteria together, the recombinant plasmid, right? So there's some plasmids, there's some bacteria, there's some plasmids, there's some bacteria. And then you shock the bacteria with temperature, or you shock them with some electricity, and it temporarily opens up some pores in their cell wall, which allows the plasmids to enter the bacteria. That's how that process is called transformation. Okay, so we're done with that example of how transgenic bacteria are produced. Now for the rest of it, you can actually read your textbook to find out a bit more about these points here on the slide. And in the next video, we'll be taking a look at other examples of genetic engineering in other organisms. I'll see you there.